theme of this term is mushrooms. And more to the point, how to grow mushrooms in a laboratory setting for a lot of different experiments. We've been learning so much as a society on how we can use mushrooms for bioremediation. We're also looking at how mushrooms can be used to make materials like tabletops, chairs. There's so many applications that we can use fungi for. So we're teaching students lab techniques and what they can do. So currently, they're in the classroom right now looking at some research opportunities, looking at some experiment opportunities on what they can do. Earlier, I was showing the students inside the mushroom cap where the white mycelium is that we can use when we clone and put them in petri dishes. Not too long ago, the students were inside the lab over here and they were looking at how to pour agar-agar. We made the recipe, so we poured it out, we measured everything, put them into the autoclave, which is a fancy word for pressure sterilizer. That way it can kill any sort of contamination so that the mycelium will have an opportunity to grow exclusively.
This is all part of a high school science class. The students receive a science credit for this class and it happens 3 days a week. We spend about 4 hours a week in this class, and the goal of the class towards the end is to have students do projects where they're able to create materials like this or grow their own mushrooms, cook with them. And so each group is going to do a new and different project and also learn some basic lab skills along with it. We have a biomaterials curriculum where students can learn about different materials that contribute to sustainability, and we believe that Green School is leading the path to take this curriculum forward.
So there's a lot of opportunity here for the students to learn some really good techniques. So when they go to university, they'll go in with an understanding of how to do things in a really scientific and proper way.